Well, thank you very much. That's a really nice introduction, and I think I have to say, go blue. That part was true, at least. I don't know about the rest. There's our slides. Thanks for the opportunity to give this plenary. It's really an honor. I'm sure I don't deserve to be here, but thank you. I want to thank also both Preston and Chris, who've helped me a ton getting ready for this talk. I really couldn't have made as much progress with it. I think I'm still working on it as I have up to this point. And I finally want to thank also the many people I reached out to in preparation. So that includes people at UNC, people around the world, really. And without fail, they're always completely generous with their time and, more importantly, their data, which I'll be sharing with you today. So thank you, everybody. I shouldn't have. You may have noticed flipping through the program that there's kind of a theme between not only the plenary today, but also the plenary tomorrow by Bonnie Ramsey, and that's A Road to a Cure. Okay, and I think with this first slide, it's been done a little bit already, but I want to take a moment for us to really appreciate the moment that we're in. I think it's almost become routine that people in CF, such as myself, talk about curing CF as if it's almost not a big deal. It's not if we're going to do it, it's just when it happens, it's going to happen. And I think, as we step back and think about it, that's something that should still make our heart race a little bit. It's a pretty incredible thing. First of all, just thinking about the impact that's going to have on our patients. We're going to take these people who really battle with their disease day by day, fight their symptoms, and we're going to be able to take that away. We're going to be able to give them back all the years of life that they may have lost otherwise. So what an impact. It's also an incredible accomplishment. Curing something in medicine is incredibly rare. We're pretty good at fixing a pneumonia or a sore throat, and surgeons can cut something out. But actually curing a genetic multi-system disease, that's something incredibly special, and we have to really hang on to the fact that we're doing something special. So as I was preparing for this talk, particularly over the summer, and thinking about this whole concept of curing CF, the phrase that kept coming through my mind was the audacity of hope. And you may have heard that phrase before somewhere else, and it's probably not a good time to talk about anything political in our country. So I promise that empty right side of the slide is not going to be Barack Obama, regardless of how you feel about it, but it's actually a painting called Hope, and we've heard a lot about hope already this morning. There's a tie-in here somewhere, I'll find it. This painting is actually said to be the inspiration for this phrase, audacity of hope. As I started to look into it with Wikipedia, which is how I learn most things, the story behind this painting, if you can look close enough and see, is that what we have here is a woman, and it doesn't look like she's having a good day. She's kind of beaten down, beaten up. She's dressed in rags. She's actually blindfolded, if you can look up here. I'm sure you can't see it, but she's holding this little musical instrument called a lyre. And if you look really closely, there's only one string left. And so this woman is in tough circumstances. But the story is that despite these circumstances, she has the audacity to remain hopeful and play music to her God. So this is a woman that's been through it. I think this is appropriate to our journey in CF is because I think it's exactly that same kind of hope that you've already heard about at the start of this session. It's that hope that continues to drive us toward a cure. You think back to maybe 10, 12 years ago, it's a time when we had the CF gene in our hand. We thought gene therapy would already have cured the disease. I think it would have been really easy at that point in time to say, well, we'll just keep plugging along, doing what we're doing. We're going to keep finding therapies that are going to make people feel better a little bit at a time. But, it's that kind of pure hope and confidence that we're going to do something big that led big decisions to make big investments to actually invest in cures and finding medicines that are going to change CFTR function directly and bring a cure to patients. So I think it's incredible what people with the right foresight and the right brain did at that time. So congratulations to all of those people. And so now that we're in the middle of this drive to really find medicines that fix CFTR and therefore cure CF, my assignment today is really to try to describe what does it look like as CFTR function begins to be switched on. And I wish I had Chris's slide. That's the perfect one. And I think this is really important because this is actually going to provide us the guideposts that tell us that we're in the right direction. So if we know what's missing, And we can start to see it come on. We're going to know we're on the right path, and ultimately, it's going to tell us when we've actually arrived at our goal. I can tell you, I'm a father of 4. They're kind of wild and crazy kids most of the time. And when you're on a long, complicated road trip to get somewhere, it's incredibly important to know where you're going and when you've gotten there so you can get out of the car. So the path I'm going to take through my talk today is, first of all, to look back briefly at kind of how we got to this point right now. And then I want to define the problem that we're facing. So what is really special about CFTR? What does it do in health? What goes wrong in CF disease? And then with that knowledge, then begin to discuss what do we measure so that we can know that we're on the right path using both in vitro techniques and, importantly, in vivo techniques, and how that'll move us forward. So in my mind, this is one of the important observations that was given to us in the 40s, really at the beginning of all this. And it's a real tragedy to think about infants dying of CF in their first days, weeks, and months of life. But that was the reality in the 1940s. And as big a tragedy as that was, it gave us an opportunity to really have a look at how lung disease develops. And what we learned from this autopsy study is that these babies with CF dying mostly from GI complications had normal lungs at birth. And the second thing we learned from this is that the earliest pathologic lesions we could see were these very bland mucus plugs inside airways without inflammation, without infection. And this was really consistent with what was seen in other organs as well. So this told us already in the 40s we knew we had a big mucus problem. I'm going to fast forward a few decades after that to the early 1980s. And I think it was this work with epithelial ion transport that gave us perhaps the reason why we have this mucus problem. So in this first couple slides is work done by Mike Knowles and Rick Boucher back at UNC. And they were interested in ion transport. They were becoming very interested in cystic fibrosis. And they already had an appreciation that we don't just want to do things in vitro, but we actually want to move these kind of measurements in vivo. So I think they were way ahead of their time at the time. And so they developed this nasal potential difference technique. And so this is a technique where you can measure ion transport in a person in their nose. And what they discovered is when looking at a normal subject versus a patient with CF, there was a big difference that was very apparent, where the potential difference or the electrical activity was very different in CF. And they were able to tease apart that this difference in the baseline measurement was due to having too much sodium absorption. And then shortly thereafter, they went and they realized there was a second problem as well, that there was an inability to secrete chloride in the CF epithelium. And that's shown here by this flat tracing in a CF patient, as opposed to this nice response to a drug that increases cyclic AMP in the normal subject. Right about the same time, Paul Quinton was working with sweat ducts and again understanding the ion transport capacities and activities in CF versus normal sweat ducts. And he came to the very same conclusions. In CF, there was a big difference in the ability to reabsorb chloride. And so together, all of this data told us that CF is really a disease of ion transport. And it gave us really the first explanations that haven't changed much over the years, that abnormal ion transport causes dehydration of mucus secretions. That's probably why we have this mucus problem. Now, probably the biggest, I think, achievement and milestone in CF research was finding the CF gene. So here we have the three amigos, Lapchi Choi, Francis Collins, and Jack Reardon, who came together from different labs, decided to work together, and really did incredible work to find the CF gene. And that's been so important because with that gene, we now have the protein in hand as well. And it nailed down the fact that this protein is a chloride channel that's also able to secrete bicarbonate, and it's taken us forward many, many steps from there. And so with the protein in hand now, it becomes very important as we think about fixing broken protein function to understand the biology of the protein. And so this may seem like a bit of a strange slide to show when discussing CFTR biology, but I was actually inspired on my summer vacation. Preston and Chris and I were having discussions, and I was getting ready to leave for a few weeks, actually, and feeling a bit stressed out. So on vacation, I'd work for a couple hours in the morning, make some slides, read some papers, and then my wife and kids and I would go out in the afternoon. My wife is from the south of France, so it wasn't too bad. But we would go to little village festivals that are everywhere throughout France in August, and we would visit, and at one of these, we came across this balloon artist. And this guy, I have to tell you, I have 4 kids, I've seen balloon artists before. This guy was special. You know, usually when you come up to one of these guys, they have their 10 things that they can do. They do the poodle and the sword. But this guy didn't have a list. He just said, "What do you want?" And at first it threw me back because I had to think, well, I don't know what the kids want or what I want. And so you come up with something, and within seconds he pulls out a couple balloons, his hands start flying around, and boom, you have something incredible. And he was a special guy. But when I went back to my work and went back to working on the talk afterward, I realized that, as special as that guy was, he really had nothing on what our cells do with CFTR. Now, imagine this long string of amino acids, more than 1,400 residues long, and as it's being created, it's being folded and shaped into these very special domains of different types in the CFTR molecule, the MSD, or membrane-spanning domains, nucleotide binding domains, regulatory domains, each with their own properties, each with their own functions. And then to make it even more complicated, which it usually is, each of these domains now have to come together in exactly the right order and the right configuration for this channel to work. So it's an incredibly complicated process. And if you get it even a little bit wrong, it turns out either we have a channel that doesn't work or it starts to fall apart and the quality control mechanisms in the cell just gets rid of it. It's so complicated, in fact, that even with a normal channel, it fails most of the time. So this tells us that we have some big hurdles to get over. We're going to think about actually making mutant channels work. So we've got some work ahead of us. Now, what I've talked about already with folding of CFTR is especially pertinent when we think about the most common CFTR mutation, delta F508. So I'm reverting back to a simpler model of CFTR with, again, the membrane spanning domains here, the nucleotide binding domains 1 and 2, and the regulatory domain. And as I've already said, it's very important how these things line up and interact with each other as the channel's being formed and as it functions. We've learned through really tremendous work that I've appreciated more and more as I prepared for this talk. One of the really important interactions within this molecule as it's formed is this interface between NBD1 and the membrane-spanning domains. It's really important for that to work. In the setting of delta F508, so a missing amino acid residue, phenylalanine, that's in the NBD domain, what happens is that, first of all, we lose that interaction between NBD1 and the transmembrane domains. Second thing that happens is that the NBD1 itself becomes unstable, and probably downstream from that, the NBD2 becomes unstable as well. So really elegant work is telling us exactly what goes wrong with the common mutation. I think perhaps even more exciting is work from Gregory Lukács' lab, who's taken advantage of these techniques of understanding the delta F channel and starting to understand how therapeutics that are designed to fix delta F508 actually work. And what they've shown is that certain of these molecules that we use in the lab, as well, in this case, two compounds actually in clinical trials, as you've heard already, belong to one class of molecule that actually addresses this problem between NBD1 and the membrane-spanning domains. On the other hand, other molecules, also correctors, act differently. They actually tend to fix this problem with instability in NBD2, and yet other molecules deal with the instability in NBD1. So we've actually learned that while we have this long list of chemicals, they actually are different and they act in different ways. And perhaps the most exciting thing that you heard in last year's plenary by Bill Skach is how when we start mixing these correctors together using single agents from different classes, how they work. And so this is some data showing that, again, if we look at the effect of a single agent, here these bottom 3 rows, with VX809 being the best single agent, we get this amount of chloride channel activity. However, when we add in a second agent, we get much more than that. When we add a third agent, we get even more. So this is telling us that it's not just an additive effect of single agents. These are now working cooperatively, and we're getting more than what we would expect with single agents. So very exciting as we think to the future of new agents for treating CF. So unfortunately, CF is complicated by the fact that it's not just one mutation we have to deal with. As Bob pointed out, there are lots of mutations, and the goal is to treat all of them. this is a diagram that tells you how we begin to classify all these many mutations into different classes, depending on where the mutant protein winds up in the cell. But I'm going to keep it a little simpler because you're going to hear a lot more about this tomorrow in Bonnie Ramsey's plenary session. And I'll keep it simple, just talking about there are those mutations that lead to the complete absence of CFTR at the surface of the cell, and there are classes of mutations that lead to CFTR that actually reaches the surface of the cell but doesn't function very well. And for the purposes of this talk, I'm really going to be concentrating on just two mutations, a delta F508 mutation, which belongs to this class 2, and G551D mutation, which belongs to class 3. So the amazing thing, given the incredible complexity of the problem that we're trying to deal with, is that we're actually having success, okay? And this is the result of high throughput screening of compounds to find chemicals that actually do one of two things. The first class of compounds are what we would call potentiators. So these are drugs that act by actually opening these mutant channels that are already at the cell surface. And so this would be attractive in a mutation like G551D that's at the surface but doesn't open and close normally. The second class of compounds we term correctors, and I've talked about them a bit already, but these are drugs that increase the trafficking or movement of mutant CFTR stuck inside the cell up to the apical membrane. And so examples of these drugs include VX770. You've heard about it already. This is the drug that came to be known as ivacaftor or Kalydeco, and is actually already approved for patients with the G55D1 mutation. And then there are two others currently in clinical trials that are correctors, VX809 and VX661. Now we know in delta F508 patients we actually have two problems. We have this trafficking problem, but we also have a gating problem. So even if we can get this molecule to the surface of the cell, it doesn't open and close very well. So the current approach is actually use combinations of these drugs. And you're going to hear a lot more about that clinical development pathway tomorrow at Bonnie's plenary session. And so are we there yet? I think clearly not. We know we have a lot more work to do all across the board. And so what are the guideposts that are going to let us move forward from here in a rational and clear fashion? I think one of the guideposts that can help us is really understanding what the essential CFTR functions are, learn how to measure them, not only in vitro but in vivo as well, then ultimately use that to figure out how much activity is enough. Clearly, CF is a complicated disease. There are many manifestations clinically. But I'm going to make the case that there are actually just 3 organ systems that lead to the majority of morbidity in this disease. And therefore, we really need to focus on the function of CFTR in these organ systems if we're going to really focus and find the right activities. And that's the pancreas, the intestine, and the lung. And I think with this kind of focus on these organs, that's also going to direct us to find the right endpoints for measuring effects to CFTR modulator therapies. And so I'm going to first talk about the CF pancreas. And these slides are from Michael Gray. All the CF slides or pancreas slides are from him, and I thank him for that. You see here a very ugly CF pancreas full of cysts and fibrotic and digested. And so what is CFTR doing normally in the pancreas? When we look at the pancreas, it can actually be divided into 2 different zones. One, the acinar zone, which is responsible for making digestive enzymes, and then ducts, which are essentially responsible for moving those enzymes out of the pancreas to the gut where they digest our food. And we know that these ducts are also responsible for making a large volume of bicarbonate-rich secretions, as you see here. So where is CFTR in this process? We know that it's localized primarily to the luminal surface of these ducts. So this is where CFTR is. And so these are cartoons that tell you the normal process and then what goes wrong. So again, we have these pancreatic ducts, we have the acini, and what CFTR is doing is it's responsible primarily for the secretion of bicarbonate, and that allows the normal transport of digestive enzymes out of the pancreas to the gut. And CF, what goes wrong is that we lose the ability to secrete bicarbonate from these pancreatic duct cells. That actually leads to blockage of the pancreatic ducts with mucin-rich secretions. And this blockage prevents the movement of digestive enzymes out of the pancreas, and the pancreas winds up digesting itself. instead of your lunch. So let's summarize what we know about the pancreas. Well, I think it's very easy to really pin bicarbonate secretion as the main function of CFTR in this tissue. As we look forward then to restoring CFTR function in the pancreas, what do we need to think about? What do we have to look forward to? Well, first of all, I think it's clear that likely to do this fully and effectively, we're going to need an early intervention, since most of our patients lose pancreatic exocrine function very early in life. However, we can't forget that 15% of our patients are pancreatic sufficient. So they have enough pancreatic function to get by, and many of these patients actually suffer from chronic pancreatitis, which can really be a horrible complication of this disease. And these patients may really benefit from this therapy as well. And I think, although it's less clear, that there may be other benefits as well to restoring bicarbonate secretion, correcting the pH of the gut lumen through that patients, even after they're insufficient, may benefit as well. What about the endocrine part of the pancreas that makes insulin? I think there are certainly potential for benefit is there. And in fact, it's the topic of plenary session 3, so I'll save all of it for them. So what about the intestine? We know that CFTR is abundantly expressed in the cells that line the small intestine. And once again, we know its job here is to serve as a bicarbonate and chloride secreting pathway. So what goes wrong in CF in the intestine? Well, in fact, when we think about it, it's pretty similar to how we think about lung disease. So in the absence of normal ion transport, bicarbonate and probably chloride secretion as well, mucus changes, and we wind up with this thickened layer of mucus between intestinal crypts and overlying the surface of the epithelium. And this in turn leads to changes in the gut microbiome and in fact frank small intestinal bacterial overgrowth. This in turn is probably a major stimulus that leads to inflammation of the gut and even frank mucosal injuries that have been shown with pill camera studies by Michael Washansky. And so this series of events ultimately leads to the clinical manifestations that we're familiar with. Patients with daily symptoms of discomfort, malnutrition, maldigestion, so really important changes. And to really delve deeper in how this works in the gut is work by Gunnar Hansen's group. And they've really taken great advantage of the CF mouse. Now, when the CF mouse was created, the hope was this would be a great model for CF lung disease, but in fact, it's turned out instead to be a great model for CF intestinal disease. CF mice don't have pancreatic insufficiency. They have pretty normal lungs. And so, in fact, we have sort of a single organ model of CF intestinal disease, and they do have severe CF intestinal disease. And so what's the basic observation? Well, on the left here, you can see a comparison of a wild type and a CF section through the intestine, and stained in green here is mucus. And we can see that the amount of mucus is greatly increased between the villi and on the top, on the surface of the intestinal surface. So there's more mucus lining the gut. We also know that the mucus doesn't behave normally. So when you prepare tissues for electron microscopy, as was done here on the right, we say that normally the mucus just floats away, it's gone, and you don't see it in this type of study. However, in CF, I hope you can appreciate it, the mucus stays attached through these preparative steps, and so it's clearly very different. So getting, again, even further down and investigating the hypothesis that it's the bicarb secreting role of CFTR in the gut that's responsible for these changes in mucus, Gunnar's group has developed this experimental system where they mount intestinal tissues in these small Using-like chambers. In these chambers, they can control the flow of fluid over both sides of the tissue, and they actually can probe the depth of the mucus layer to measure it directly. I'm going to show you a couple movies that he provided for me that really demonstrate what's going wrong in CF. So on the left is first a movie from a wild type piece of small intestine, and I'll point your attention to these little black flecks. These are little carbon flakes that are just there so you can see the mucus that otherwise is pretty clear. And so we'll get the movie started here, and you'll see a pipette coming in, and the experimenter is merely going around sucking the mucus off of the cell surface, and within seconds it's gone. Okay, what happens in CF? You see the pipette come in, moving around the tissue preparation, moving around the tissue preparation, sucking. And at the end of this, we're left with a tissue that still has mucus on top of that. Okay? And this is quantitated on the right here, where we see kind of the before and after of attempting to move that mucus layer. In a normal tissue, mucus goes away. In CF, nothing is different. It doesn't change. So clearly that mucus is behaving differently. And then to get again at that hypothesis, is it the bicarb secreting role through CFTR that's important? What was done is actually first taking a normal small intestinal tissue, mounting it, and then make it CF-like. Take away its ability to secrete bicarbonate, in this case simply by removing bicarbonate from the backside of the tissue. And what happens in this normal tissue is that it now becomes CF-like. And so the remaining amount of mucus left on top of the tissue is much thickened, much like in CF. And the opposite experiment was done as well. So here we're taking CF tissue, mounting in the chamber, and then now giving extra bicarb to the apical or luminal surface. And we can see that as additional bicarbonate is added, now the CF mucus behaves more normally, and it can be removed from the surface of the epithelia. And so to summarize the intestine, it's very clear that CFTR is incredibly important, especially for bicarbonate secretion once again, and that when we don't have that, we're left with sticky mucus, or mucoviscidosis, as Paul Quinton reminded us in his important publication recently. And that what happens after the development of this abnormal mucus layer is very reminiscent of what happens in airways. Mucus, infection, changes in bacteria, inflammation, and injury. I think it's exciting also is that it's very clear that we can expect big improvements in patients as we restore CFTR function in the gut. And this comes from very nice work done whereby CF animals, so both CF pigs and CF mice, have had their CFTR function only in the intestine genetically restored. And in these animals, we see very clear and significant differences in the phenotype of the intestinal disease. So for me, that tells me that if we can do the same thing in humans with medications, we're going to do very good things for the intestinal manifestations of CF as well. Okay, lastly, let's talk about the lung. Again, this is what I deal with primarily. We know that CF is a disease that over time results in accumulation of mucus in the airways and very severe infection and inflammatory process. I think as we've, many people in the field have worked very hard to figure out exactly what goes wrong in CF. When CFTR is missing, why do we develop this disease? And I think one thing we can all agree upon is that there is this cycle that happens. When airways become obstructed, that predisposes to infection, and that in turn leads to an inflammatory response. But the exact role that CFTR plays in this is not completely understood. Is it playing a role by altering ion transport and mucus properties such that the airways get obstructed primarily? Does it have a direct effect on infection? Or is it the loss of CFTR causing dysregulation of inflammation? I think there are a lot of questions still open to this. But there's been really significant advances very recently that give us information on how this works. So this is work done by Brian Bunton, working in Rick Boucher's group at UNC once again, where they really challenged our basic notion of what the system looks like on airway surfaces. So we've been looking at pictures like this for a long time where we see airway epithelial cells. These are cilia surrounded by a clear space and then mucus sitting on top. And the basic notion has been that these cilia are beating in a watery environment, makes it very easy, and that allows mucus to be essentially propelled out of the lung. And so this is the model. We have CFTR and ENaC controlling how much liquid is there, and mucus kind of magically floating on top. But as this model was really looked at, including by groups of people from different walks of life, physicists, gel polymers, and what became to be known as the virtual lung group at UNC, many questions began to arise that really brought into question whether this is reality, including the basic fact is why doesn't the mucus encroach, kind of sink down between cilia and prevent the ability of cilia to beat and propel mucus? So instead, an alternative model was proposed, whereby instead of a watery layer around cilia, in fact, there was a second gel. This second gel would be comprised of different mucins that are secreted and up in this overlying mucus layer, but instead tethered mucins. So these are a class of molecules that remain attached to the cell membrane and essentially are out in the space in between the cilia. Because these tethered mucin molecules are covered in negative charges, they don't like to be around each other, they repel each other, and essentially form like a bottle brush around these cilial shafts with mucin molecules projecting out from that. And this does a lot of nice things. First of all, that's a wonderful low friction environment for cilia to beat, but it also gives you the barrier function that we know is there, and it also keeps this floating mucus layer out of the PCL. Now, it's a great theory. We also want to have evidence that it actually exists. And so this is actually work done at UNC as well by Camille Hervé, who also, in full disclosure, happens to be my wife. These are electron micrographs. I should have her come up and tell you. Electron micrographs of this space. And so here we're really focusing in on a very small area between cilia. We see the edge of cilia here, and we can see that this space doesn't look like a simple watery area. There's structure to it. Okay, so that's our first hint that something's there. And then Camille came in with antibodies directed against specific tethered mucins, MUC1 and MUC4, and saw that indeed there's very strong evidence for these molecules at specific locations in the periciliary space, MUC1 being down here close to the cell surface and MUC4 being distributed more evenly throughout the PCL. And so instead of this gel liquid layer, we're now left with a 2-gel model. And so how does this relate to mucus transport, the thing that's probably most important to what we're talking about? Well, if we're thinking about this normal system here as being comprised of 2 gels, these 2 gels are, in essence, a softer gel on top and a harder gel down below. And this harder gel will have the propensity to suck water into it because of its higher osmotic pressure. However, because it's tethered to all of these structures, it can't grow very much. It's fixed to a confined space. And so it's going to stay pretty much as it is. And any extra water that's in the environment will just stay up here in this secreted gel layer. However, in CF, ion transport is driving the progressive dehydration of this layer. And what will happen initially will be well tolerated. All the extra water that might be in that overlying mucus layer will be absorbed. And that can happen fine for a while until we really reach a critical threshold or tipping point where the osmotic pressure in these 2 layers, the overlying gel and the PCL gel, are equal. Any further absorption after that means that water is going to leave both layers, and now we will have encroachment and deformation of that PCL layer. We're going to have adherence of mucus to the cell surface, and we're going to lose the ability to transport mucus. And so what does this mean very briefly? It means that CF mucus is going to be dehydrated, and it's going to have measurable elevations in osmotic pressure. And that beyond a specific threshold, the PCL gel is going to become deformed, mucus is going to become adherent to the cell surface, and mucus clearance will fail. I think it's a nice model also, in that it allows for a progression of disease over time, that this can be maintained for a while, clearance will remain normal early, but then over time we're going to have the accumulation of heterogeneous defects throughout the lung. That is very attractive as we think about how lung disease progresses in CF. Now, the second area that I think is incredibly exciting and expands our knowledge of lung pathogenesis comes from the CF pig and the Iowa group. We know that the CF pig really behaves a lot like humans with CF, in that they develop mucus filling airways, mucus attached to epithelial surfaces. But these pigs also provide a wonderful large animal model to explore something else, and that's the direct ability to kill bacteria as a function of CFTR. And so the group in Iowa developed this really elegant experimental system where they were able to attach bacteria to this grid and then carefully lower that grid down onto the surface of cells and then assess the ability of that airway surface liquid compartment to actually kill the bacteria. So after removing the grid, all they had to do is look under a microscope, use special stains that tell you whether a bacteria is alive or dead, and count up the cells. So this is what the system actually looks like. This is the grid to which bacteria are attached. And And then this is what the grid looks like under the microscope after staining for live cells, which are green, and dead bacteria, which are red. And so the really fundamental observation from these experiments is that when you lower bacteria down into this airway surface liquid on a grid, we're taking out clearance of bacteria from many mechanisms, and we're really solely looking at the ability to kill those bacteria directly. And we see within really 30 seconds and a minute, there's a striking difference between CF and normal. And normal, in the range of 75% of those bacteria have already been killed by 1 minute, whereas in CF, the amount of killing is dramatically reduced. And so the question is it that there's not enough killing molecules, antimicrobial molecules, or is it that they don't work very well? And I don't have all the evidence that the Iowa group presented, but they did very beautiful work that really convincingly tells us it's not a lack of antimicrobial molecules in this space, but it's really their function. And so they went on to pursue the hypothesis that, once again, it's perhaps CFTR function as a bicarbonate channel and a regulator of pH that could be a result for this killing problem. And so the first step of this, obviously, is showing there is a pH difference between non-CF and CF pigs, and indeed they showed that. And then they went in to then link this difference in pH to bacterial killing. And they did this first by actually taking normal or non-CF pigs. They exposed them to CO2 in the environment, and this, as it predicted, did lower the pH of the airway surface liquid layer. And in conjunction with this, when they measured the ability to kill, we see that indeed killing of bacteria in this now more acidic environment was impaired. The opposite experiment, once again, was done as well, taking a CF pig, exposing it to inhaled sodium bicarbonate, which, as expected, raised the pH of the airway surface liquid layer, and this in turn translated to improvement in killing. So very strong evidence that CFTR's function in the airways may also be to regulate pH of airway surface liquid, which in turn impacts bacterial killing. So I think we've come a long way recently in our understanding of lung pathogenesis. And now I think we're at a point where I think it's okay to accept that CFTR may be doing more than one thing, affecting mucociliary clearance and mucus properties, and perhaps also affecting bacterial killing directly. I'm going to leave a dash line next to inflammation as well, as it's also very possible that CFTR has a role in regulating inflammation. So I've outlined what I think are the key functions of CFTR that lead to the important parts of CF disease. And so now, as we now move to trying to impact the function of CFTR, we need good assays that lead us along the way. The drug development pathway goes from high-throughput screening and compound identification, optimizing those molecules, and then getting to this part of the process where we need to pick the ones that go to humans and ultimately decide if they're working or not. So what we really are asking for our assays are that, first of all, they quantitatively reflect CFTR function, and second, that hopefully they're going to predict therapeutic success. And so I want to start out by talking about a couple in vitro assays that are extremely exciting. I think will turn out to be incredibly important in this process. And the first of these are the intestinal organoid system. And I'd like to thank Jeff Beekman and Joanna Deckers, who provided these slides and these data to share with you. Intestinal organoids are actually obtained by taking a very small biopsy from a patient's rectum, using those cells, putting them in very special culture conditions, which allowed them to massively expand and ultimately form these little, somewhat spherical, hollow balls, we'll say. And these little structures very nicely express CFTR, as you see here, on the luminal surface of these structures. And what I'm showing you here is what these things look at somewhat lower power. Here we have a whole mess of organoids, in this case from a control tissue, and next to it from a CF tissue. And what the Beekman group is going to do is expose these organoids to forskolin, which is a chemical that raises cyclic AMP, which should turn on CFTR if it's there and if it's functional. So we're going to see this little movie from the wild-type organoids, and I hope you can see that they swell very nicely. So CFTR is being turned on, liquids being secreted inside the organoid, and the whole structure swells. What happens in CF? I promise you the movie's moving, but not much happens because CFTR isn't there. So this gives us a really nice assay to quantitate CFTR function that clearly discriminates between CF and wild-type tissues, and also can discriminate between more intermediate function as well. So actually, if you even just look at the baseline size of these organoids, you can see that in severe CF mutations, mild CF mutations, and in control tissues, the baseline size is actually a little different. But more importantly is how the swelling of these organoids perform and how they compare to assays of CFTR function from freshly obtained tissues measured right out of the patient in an oosing chamber. And as you can see, they match up extremely well. So whether it be CFTR mutations other than delta F508, severe CF from delta F508, mild CFTR mutations here, or control, they match up very well, telling us we lose very little by moving from this freshly excised tissue, which is a very limited resource, to one where we can massively expand it and study it in culture. So a wonderful system to help us move forward. The other really beautiful thing about the organoids is that they seem to give a very nice readout on CFTR modulator therapies. And so here, in a collaboration with Gergely Lukács, is the effect of single and multiple corrector compounds on the swelling of these organoids. We can see here first is the effect of VX-809 by itself, again, the best single agent that we have currently, the effect of adding 2 drugs from 2 different classes, and the effect of adding 3 correctors from 3 different compounds. And then this fourth bar is in the presence of a CFTR inhibitor, telling you that all of the swelling is completely mediated by CFTR. And I'd like to also point out here that the actual results obtained in these experiments are once again, much larger than the predicted results that we would guess from just adding up the individual responses. So telling us that we really have to have great hope for how drugs from different classes are going to work together. Now, I'm a pulmonologist. What's done in rectal tissues is wonderful, but I would like to do the same thing in lung tissues as well. Clearly, we don't want to miss the boat on studying compounds that might work very well in one tissue but not another. And the capacity to do this is actually coming online right now as we speak. And this is work done by Dick Schlegel at Georgetown in collaboration with Scott Randell at UNC. In these experiments, we have the ability to sample, again, very small tissues, either with a nasal scrape or nasal brush, and then culture them in very specialized conditions with a Rho kinase inhibitor, this Y compound listed here, and in the presence of fibroblast feeder cells. And in these very specialized conditions, the cells go from a differentiated condition to actually a stem cell-like condition where they begin to expand like wildfire. They double, they double, they double, they double. You can see them, these doublings occurring in these specially treated cells as opposed to what happens in our conventional culture conditions. Then the beautiful thing is that when we take these cells that have now massively expanded, then put them back into normal culture conditions, it turns back into a beautiful airway epithelium with cilia here in white, goblet cells in green. Looks just like we would expect an airway epithelium to be. And then this now expanded epithelium is available for whatever your high throughput assay of choice is. And so when we now consider both the organoid system and this Rho kinase treated airway epithelial system, I think there are some important implications. The first of all is this gives us resources that from a single patient, we may be able to test multiple drugs, test multiple doses of drugs, test drug combinations to find what works best. And I think Bob really did a great job of actually pointing out how important this is as we think about wanting to treat patients of all mutations, not just the common ones, but of all mutations. This is when we use our common CF screening mutation panel. It contains 23 mutations. How does this line up to the total number of mutations? Well, it's about 1%. What about CFTR2, this wonderful new tool and database that's available to us, well, that includes 160 mutations. And while that covers most of the patients out there, it's still only about 8% of the total number of mutations that have been reported. So these techniques, I think, really give us now the possibility for personalized medicine, being able to take a tissue from a patient, massively expand it, and do whatever we need to do, testing different compounds, combinations of compounds to find out what works for that patient. So a wonderful, exciting possibility. So I've talked a bit about in vitro assays, and I want to transition to in vivo assays as well. And it's always important to make that transition. Ultimately, we're going to be studying patients, and we want to know when our drugs that are affecting CFTR function, whether they're working or not, and how well they're working. I think it also gives us the opportunity to not just study a channel or ion transport from a channel, but it gives us the opportunity to really study a physiologic system that's impacted by CFTR. And so we're not starting from scratch, thankfully. And in fact, I've mentioned one of these assays already, the nasal potential difference assay, which has been very important and used very well in our early studies of ivacaftor for G551D. And Steve Rowe has done a really good job going back to these data and really looking at how does this assay perform. And you can see that in the case of ivacaftor treatment of G551D, we saw a treatment effect of about a 5 millivolt change in chloride secretion. And that certainly is large enough to conduct a study of a reasonable size and get a result. However, as we think forward to testing agents that might not be quite as effective as ivacaftor for G551D, and we start having smaller treatment effects, you can see that the number of patients we need to study just balloons, and it may quickly exceed the feasibility of doing a study of this size with this very specialized technique. What about sweat chloride? Once again, sweat chloride has been a wonderful assay to tell us that a drug is affecting CFTR function. We see that from the phase 3 trial of ivacaftor and G551D, as well as the GOLD study that's been presented here at the meeting already. Huge effects on sweat chloride. But there's some problems with this assay as well. We can see that in this publication, that was actually put out by the FDA, that there's not a relationship between a change in sweat chloride and a change in lung function within an individual. It's a bit of a scattergram. So what this tells us is that we can't use sweat chloride to predict the clinical response in an individual patient. I think there's some other issues as well. So once again, when we looked at ivacaftor treatment of G551D, we got a big response, both in lung function and in sweat chloride. However, as we've begun to accumulate data on combination therapy and single drug therapy in Delta F508, we seem to see a different relationship. We're actually seeing some nice lung function responses in the phase 2 trials without much of a change in sweat chloride. So it seems that the relationship between sweat and lung function are different. And whether it's because we're moving from one genotype to another, or because we're moving from one drug to a different drug, we don't really know, but it's an issue. And it calls into question and reinforces our need to have other assays as well to assess CFTR function. And so this problem and this issue led to the decision to conduct this GOLD study, the G551D observational study. The hypothesis underlying this study is that we have this opportunity. We're going to be turning on CFTR with ivacaftor in patients with G551D, and with this restoration of CFTR activity, we may be able to see and identify outcome measures that are closely tied to CFTR function. And so let's think back a few minutes to what I described as perhaps the most essential functions of CFTR in vivo, in the pancreas, bicarbonate secretion, in the ducts, fixing intestinal pH. That'll affect mucus layers, et cetera, and mucus transport in the lung, and regulation of ASL pH. And so it raises many things that we might measure. But what did we measure in GOLD? Well, the first thing we measured is the intestinal pH, reflecting bicarbonate secretion in the pancreatic ducts. We measured mucociliary clearance, again, assessing this hypothesis about mucus hydration and clearance in the lung. We measured bacterial infection, which could be the result of a direct effect on bacterial killing, or perhaps would be a downstream effect on mucociliary clearance. And we measured inflammation. And there are other things as well that got measured, but these are the key ones I want to talk about today. So this was the overall design of the GOAL study. Patients were enrolled, they were followed for 6 months, and in all patients, kind of classical clinical trial outcome measures were collected: lung function, patient-reported outcomes, as well as sample collection for future biomarker work, serum, plasma, DNA, etc. But nested within this overall study were 4 sub-studies that you see listed here, and I'm going to talk about them, 3 of these in a bit more detail, but want to refer you to Symposium 10 and these posters as well for more information about both the overall study and the sub-studies. So let's talk first about the intestinal pH measurement study. And so this utilizes, it was led by Daniel Gelfand and Drusi Borowitz, who you've already seen today, but utilizes this very nice technology where, in fact, a wireless capsule is swallowed by the patient. It moves through the digestive tract and reports back pH, motility, and temperature wirelessly to a data recorder. So the underlying hypothesis is that intestinal pH will be low in CF, and that ivacaftor is going to correct that in G551D patients. And so some earlier work prior to GOAL by Daniel Gelfand shows us that indeed intestinal pH is abnormal in CF. So here is a tracing from CF patients, and here's a tracing in healthy controls, seeing a clear difference, especially in the earlier part of the intestinal transit with lower pHs in CF. There's a couple dotted lines here as well that I want to bring your attention to. This is at pH 5.5 and 6, and this is the pH at which pancreatic enzymes start to work. So it's very clear that early in the test, particularly, pancreatic enzymes aren't going to work very well in CF. So what happened in the GOAL study? Once again, we see that in prior to the start of ivacaftor therapy, this black line, we see that pH was again very low for an extended period of time, but after initiating ivacaftor therapy, pH was corrected. So it rapidly got up into this zone that we want pH to be for enzymes to work, and it actually remained higher throughout the entire transit. So I think there are a lot of clinical implications that come from this kind of data. First of all, it's possible that correcting CFTR function is going to help exogenous pancreatic enzymes to work. I think it's very likely that these improvements in intestinal pH may be responsible for the improved GI symptoms and nutrition that were observed in the GOAL study and in the phase 3 studies of ivacaftor. But then, once again, if we're going to preserve pancreatic function, we're going to need to do this early in life. The second sub-study that I want to address is the mucociliary clearance study. The underlying hypothesis of this study is that if indeed CFTR is closely associated with the control of mucociliary clearance, that treating patients with G551D with ivacaftor should yield significant improvements. And so in this study, we enrolled 22 patients at 4 study sites, measured mucociliary clearance at baseline, and then 1 and 3 months following the initiation of therapy. And I think this is really an important sub-study because it does get at this hypothesis of pathogenesis. Once again, if defective CFTR really is tightly linked to altered mucociliary clearance, we should see it in this study. On the other hand, if abnormal mucus clearance is more the byproduct of having an injured airway downstream from these events, I think it's fairly unlikely that we wouldn't see a difference in mucus transport. So for those of you that might not be familiar with these techniques, in concept it's fairly simple. Patients first undergo what we call a transmission scan, which is kind of like a really cheap x-ray that allows us to identify the boundaries of the lung and also to define regions of interest. So here a central region of interest and here a peripheral region of interest. They then inhale radiolabeled particles in a very defined fashion so that we can get reproducible deposition of those particles in the lung. And then they sit in front of a gamma camera and we take serial images over time so that we can quantitate the disappearance or clearance of these particles over time. And so this is an example, a really nice example, of what can happen with ivacaftor therapy in a patient with G551D. I have a couple landmarks to point out to you. First of all, this is the stomach. So these are particles that likely hit the back of the throat and got swallowed. So these little hot spots under the lung are stomach. And then I want to draw your attention also to this area above the lungs and in the middle. This is the trachea. So it's possible with mucus clearance you may see signal moving out through these areas. So let me play the baseline video first. And to the trained eye, there's probably a little clearance going on, but it's not real exciting. And then after ivacaftor, it's pretty cool, right? All the particles are leaving the lung in a rapid fashion. You can see it streaming up through the airways, likely doing very good things for this patient. I want to, again, thank Tim Corcoran for making these videos out of our data collected at his site. And so it's not just an N01 that looked really good. It's actually all the data that looked really good. So this is the average whole lung clearance from patients at baseline in black, and then after 1 and 3 months of therapy in the red and the blue, respectively. So you can see a clear difference here. We typically look at these clearance curves by calculating average clearance through 60 or 90 minutes, and that's shown on the right here. And so you can see very clearly the clearance has essentially doubled on average after the institution of ivacaftor. I want to also show you data that was from the peripheral lung. Now, we're really interested in what happens out in the peripheral lung because this is where most of the small airways are. We really believe that in CF, this is a key site of disease, both the initiation of disease and the progression of disease. And if we now put your attention to the clearance curve from this region of interest, we can see that there's really not very good clearance under baseline conditions, but after 1 and 3 months of therapy, we see a very significant increase in clearance. And again, the summary data with average clearance on the right, seeing a 6, 7, 8-fold increase in average clearance in this lung compartment. Now, this is really exciting because as we've studied a lot of inhaled therapies over time, and I can tell you it's very difficult to get enough of an inhaled compound deep into the lung to impact peripheral lung clearance. Because we're now using a systemic therapy, I think that's really the key of being able to do much, much better on mucociliary clearance. What about infection? This is actually data from the overall goal study, and I think was a bit of a surprise for all of us. So what was done is sputum was collected during the course of the 6-month study, and because registry data was also part of the study, we were able to look back in time and see the fraction of patients who had Pseudomonas before the start of ivacaftor versus after institution of ivacaftor. And I think, again, the big surprise was that about 55% of patients had Pseudomonas before starting therapy, but this dropped significantly down to about 35% of patients after starting ivacaftor. And this was sustained through 12 months. Now, I think when all of us first see this kind of data, we immediately start thinking, well, where's the bias here? Where's the mistake? Where's the thing that's really pushing it wrong? And I can tell you that Steve Rowe and Sonia Helshi at the TDN Coordinating Center have done multiple additional analyses to really make sure that this is robust. And they've definitely made a believer out of me that this holds up very well when correcting for everything that we can correct for. So this is exciting. What about infection? Scott Sagal led one of the sub-studies as well, where he looked at microbiome effects as well as effects on inflammation. And I'm not going to show all of his data, but I can tell you that there's some really interesting trends on what's happening to the microbiome. And because he only had 14 patients, we're going to really work harder in an add-on study to go to get more samples so that we can really follow up on that in detail. But inflammation, unfortunately, did not show a difference, again, in these 14 samples, for either because we don't have enough samples or perhaps because we're studying patients that already have pretty severe disease. And I think it's very likely that, again, as we move therapies earlier in life, prior to the establishment of severe disease, I think this is an outcome measure that might turn out to be very important and useful. So we've talked about assays, in vitro, in vivo. How much CFTR function is enough? And we've seen these type of depictions comparing sweat chloride changes to NPD changes in patients with classic CF versus milder forms of CF. And the ivacaftor experience, once again, I think is going to give us a really nice benchmark. If we overlay the sweat chloride and NPD responses to ivacaftor, it's giving us an estimate that perhaps we have about 30% CFTR function. I have to tell you, I heard a great talk this morning already from Jeff Wine, working in the sweat gland, who came up with a very different number. And again, this is telling us there may be tissue-specific differences. And this is a difficult question to answer. And in fact, this slide, after much thought, is kind of my punt on the whole answer of how much CFTR function is enough. We've typically compared sweat to NPD and things like that, but I think what we really want to do is compare different markers of CFTR function against disease burden. So what we want on the left-hand axis is lung disease, pancreatic function, nutritional status, and then compare that to other useful markers of CFTR function, whether it be from an oosing chamber, from an organoid, from an NPD, etc. And so how do we then plot things onto this graph? Well, we know where we want to be. We want to be at 0 disease burden, so that's where we're going. This is where we are before any CFTR modulator therapies. We have all the disease burden with no CFTR function. Once again, ivacaftor and G551D gives us kind of our first data point somewhere on this graph, and it's a big circle because there's a fair bit of uncertainty around it, but we know we've made a pretty big dent in disease burden. We know that we're getting a fair amount of CFTR function, maybe 30%, we don't know. As time goes on, we're going to be able to now fill in more and more points. And I think some of the most important points are going to be the results of the phase 3 trials of combination therapy in Delta F508. I've put them here. I don't really know where they're going to go. But by putting them somewhere on this curve, we're ultimately going to be able to now begin to plot out exactly how far we've come and how close we are to this goal of having 0 disease burden and how much CFTR activity that's going to take. So we have a bar with ivacaftor and G551D. How are we going to move past that? I think we're fairly convinced we've done great things, but we're not all the way there, even for G551D. These patients still have persistent disease. They don't have completely normal sweat chloride values. They don't have completely normal nasal potential difference tracings. So even in this case, we want to do more. So how are we going to do that? I think one possibility is simply starting earlier. Again, our studies on G551D were in patients that were over 6, adults, and that's a higher bar to get over than I think if we can come in earlier before that disease is established. I can tell you that this work is already underway. So trials are underway studying ivacaftor in 2 to 5-year-olds now with gating mutations, and I think we're going to learn a lot from that. Do we do better by starting earlier? Another way that we can do better is treating both mutations. So with ivacaftor, we treated G551D. But what if we could treat both mutations? So what if you're a heterozygote, you have delta F on one allele and G551D on the other? What if we can treat not only G551D, but we can treat delta F as well? Well, that hypothesis is also about to get tested as well in a phase 2 clinical trial of VX661 with ivacaftor. So in these patients, we're going to identify patients who have exactly this scenario. They're heterozygotes for delta F508 and G551D. These patients will already be on ivacaftor for their G551D, but we're going to see what additional benefit we get by treating that delta F508 allele with this combination approach. And clearly the other way of doing better is simply having more and better drugs that are available for everybody with CF, for all mutations. And there's little doubt that we're on the right track for that because of the strong support of the CFF, from NIH-supported studies, and for the collaborative work that's being done by people in academics and by industry partners as well, of screening for better drugs, screening smarter to identify them, and moving us forward. But moving forward, there will be challenges. I think we want to continue to refine and expand upon CFTR endpoints really at all stages of development, whether it be selecting which drug goes to man or assessing the efficacy of a drug in a person with CF. I think we then want to really consistently use these tools that we develop to identify the best therapeutics and take us closer to that goal, closer to there. But there's no doubt we've made tremendous progress on knowing what to measure in vitro and in vivo. and that there's no doubt in my mind that the CFTR therapies are going to continue to get better and better and better, that we really have a clear development pathway already mapped out for us. And so in my mind, it's very clear that a CF cure is indeed in sight. So with that, thank you very much.